So in this section, we shall look at the primary and secondary structure of proteins a little bit more closely. So before we discuss primary structure and secondary structure in this section, let's first define complete proteins and its relation to nutrition. A complete protein provides all the essential amino acids. So now we introduce a new term, essential amino acids. So what are these? Okay. Essential amino acids are amino acids that we cannot synthesize, and therefore we need to obtain these from our diet. Why is it important? Well, as we will see in this chapter, amino acids are the building blocks of proteins. Therefore, if you do not have all the amino acids needed for a protein, then you cannot synthesize that protein and it will impair your ability to live. Okay, so what are these essential amino acids? We have methionine, isoleucine, leucine, lysine, phenylalanine, arginine, histidine, threonine, valine, and tryptophan. It's very important to note that not all types of food provide all the needed amino acids. Okay, this is most especially important for vegetarians because if you don't eat a variety of foods, then you may not have all the amino acids, all these essential amino acids. Okay, so let's go back to primary structure. Actually, we'll go through this very quickly because I think I said almost everything about primary structure in the earlier presentation. Okay, so it's a sequence of amino acids in the polypeptide chain. As mentioned, it may and most likely affect secondary structure, tertiary structure, and quaternary structure of proteins. If there are changes in the sequence, as we've seen, you may have negligible effects, so that's good. We've shown the structure of the protein earlier, from humans to yeast. Okay, so there can be changes in the sequence and still have the protein function as it is. In some cases, it may have minimal effects, meaning it just doesn't work quite as well as it should because of these changes. Or under extreme cases, then you get loss of activity. So this is now very important and has a very big effect because the proteins have important functions. And if you lose that, then the cell might actually die. Okay. So one example we'll look at later will be sickle cell anemia. This is caused by a change in one amino acid. We shall be looking at myoglobin in just a short while. So here's now is the primary structure of myoglobin from different organisms. Okay, so if you look, you may want to pause the video and look at where there are changes. Now, here's just 1 to 5 species from yellowfish tuna, yellowfin tuna, shark, chicken, penguin, and the sperm whale, myoglobin. Okay, so let's look at secondary structure, which refers to the arrangement of the polypeptide backbone. As mentioned earlier, there are two regularly repeating secondary structures. These are the alpha helix and the beta sheets. Okay, they're stabilized by hydrogen bonds, and the hydrogen bonds form patterns. There's one pattern for the alpha helix and another pattern for the beta sheets. So let's analyze. We expect free rotation within the polypeptide backbone around the alpha carbon. Reminder, between the carbonyl carbon and the nitrogen, okay, forming the peptide bond that forms a peptide plane, so there's no free rotation around that carbon-nitrogen bond. Okay, so what's in the alpha carbon? Well, the alpha carbon is connected to the amino nitrogen of the residue, and also to the carbonyl carbon of a residue. So a analysis of this, okay, showed Ramachandran angles. okay, as shown in this figure. Okay, so focusing on the alpha carbon towards the middle of the figure, okay, you can see that there are two peptide planes connected to that, associated with that. Okay, as shown in the top part of the figure, we have the angle phi. Okay, so there's free rotation around that bond, no? So that carbon-carbon bond is a single bond, so that can freely rotate. That is given by the angle phi. And then that alpha carbon is also connected to the nitrogen of that same residue. Okay, and it's also connected to that one has another peptide plane. Okay, so that angle is defined as phi by Ramachandran. Okay, don't worry too much about this, but again, we just need some point of reference. Okay, so I highlighted that residue, no? So that this is one amino acid residue. Okay, so let's look at the alpha helix a little bit more closely. It's a rod-like structure of the polypeptide backbone, and it involves only one polypeptide chain. So if we analyze the figure on the left, okay, I'll start with this nitrogen. This is the uh, amino nitrogen, which is now involved in a peptide bond with a previous residue. This residue is incomplete, so I'll start with this one. So you have the amido nitrogen, the alpha carbon, and then the carbonyl carbon. Okay, see, it's connected to the oxygen. So this is one residue. This orange sphere or yellow sphere, whatever this color is a side chain. And then, of course, there's a alpha hydrogen also. We ignore them. Okay, and then the next residue is composed of this nitrogen, alpha carbon, and the carbonyl carbon. Next residue, amido nitrogen, alpha carbon, and the carbonyl oxygen, and so on. Okay, so it forms a helix. Okay, so this side represents the N terminal. This side represents the C terminal for the polypeptide chain. And it's stabilized by the hydrogen bonds that are parallel to the helix. So if you look at the direction of the helix, it goes from N terminal at the bottom to the C terminal at the top. Where are the hydrogen bonds? These are now shown by this little red dots. And they have the same direction. Therefore, from your algebra trigonometry, we say that the hydrogen bonds are parallel to the helix. Okay, and then let's count. Where is this hydrogen bond? Okay, so let's look at the carbonyl oxygen of this residue. Okay, it is hydrogen bonded to this hydrogen of this amido group. But this residue is 4 residues away. Okay, so if we count, this is the residue in question having this carbonyl oxygen. This is now the first residue after, the second residue after, the third residue after, and this is now the fourth residue. Okay. This is also known as a 3.6 helix because if we now look at the helix and then see how it turns from this direction, from this position, and then it turns, and then we go to the same side, it actually doesn't complete like 4. It's not really 4 residues away. Okay, it's only composed of about 3.6 of residue to go to the same side. Therefore, again, this is like 3.6 residues away, or as described here, 3.6 residues per turn. Now let's count how many atoms are involved in the hydrogen bonded loop. There's 13, so let's count them. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and 13. So there are 13 13 atoms in the. There are 13 atoms. Sorry, I might tell them. There are 13 atoms in the hydrogen bonded loop. Okay. Another perspective of the same picture, okay, could show the helix. No? This time we're looking through the helix. Okay? So on the top figure over here, this is the a portion of the again and the alpha helix of a horse liver alcohol dehydrogenase. Okay, the identity of the protein isn't really important. Okay, but again, this is the primary structure. Okay, it just shows the sequence. Okay, so this is residue 355, 356, and so on up to 365. This section shows an alpha helix. Okay, and then it's now shown over here. No? So it just shows a spiral. Okay, so starting from the N terminal. Okay, so that's residue number 355, 356, 357, 358, 59, 60, and so on. Okay, so here you can probably appreciate more that it goes from the 3.6 turns. No, so if we start from the isoleucine over here and then make a turn, and then if we reach over here, it doesn't quite match with the fourth amino acid. Okay, so this is the amino acid in question. First residue away, second residue away, third residue away, and this is the fourth residue away. So as you go to the same side, it appears that there's only 3.6 helices, 3.6 residues involved to go to the same side. Okay, and then we'll also see that again, it's not shown over here, but we can infer from the description of the alpha helix in the earlier slide that the carbonyl oxygen of isoleucine will be hydrogen bonded to the amido hydrogen of phenylalanine. Okay, so this is the fourth residue away. So so this is just another perspective showing the alpha helix. Okay, in reality, it's like a helix, not like a spiral there. They should be superimposed on each other, except that again, it will, for clarity, it's shown like in a somewhat like an exploded view. So, as mentioned, the alpha helix. Okay, this arrangement gives maximum strength of the hydrogen bonds. Okay, so recall your general chemistry. Okay, a hydrogen bond is actually much weaker than a covalent bond. However, if you now count all these residues, all these contributions of these hydrogen bonds, then it's now able to keep the polypeptide chain in a helical arrangement, and it contributes to the strength. So it's not flopping around. No? The helical conformation is very stable, as mentioned earlier, 3.6 residues per turn. And then the pitch is 5.4 angstroms. What does that mean? Again, you go from one section of the spiral and then make that turn. And then once you reach that same side, determine the distance. Okay, so the distance is also known properly as a pitch, I guess, in their language. Okay, it's a pitch, no? So there's 3.6 residues per turn, and the pitch is 5.4 angstroms, meaning from this to this is 5.4 angstroms. Okay. So here are different perspectives of the alpha helix. Okay, so the one on the left is the ball and stick model. On the right, it superimposed the ball and stick model and the peptide planes. So sometimes you may ignore the ball and stick atoms and just look at the arrangement of the peptide planes. So the peptide planes also form helices. And then over here is the space filling view. Okay, so it's still color coded. Reminder, it's color coded. Black means carbon, blue means nitrogen, red means oxygen, white is for hydrogen, and then for the other atoms, then it's you can now probably pick and choose whatever color you want. Okay, so and then on the right is a ribbon structure. Okay, just emphasizing. Usually this is used to represent again the alpha helix itself. No? So maybe these carbon atoms, nitrogen atoms may not be even written. Okay, so which one is the accurate one? Well, it depends. What are you emphasizing in your presentation? Now, what do you want to emphasize? You're talking about the protein structure. What do you want? Okay, do you want the ball and stick model for clarity, and or do you want the space filling model, or do you want just the ribbon, or do you want to emphasize the peptide planes? Doesn't matter. Okay, so what's important, what is appropriate is the one that will be used. Okay. However, before I move on, I probably just want to say something important. Okay, you're probably used to the ball and stick model because it shows the connectivity of the atoms. However, this figure on the left is the same as this space filling model. See the difference? Okay, in the space filling model, exactly what it says. It actually fills up the space. No? In the ball and stick model, the stick that represents the bonds are actually exaggerated. Okay, so we may be deceived and state that, oh, there's a lot of space in between the atoms. In reality, there's almost none. Okay, so always keep that in mind when you encounter the ball and stick model. Okay, keep in mind that again, the bonds are probably exaggerated in length. Not probably, the bonds are exaggerated in length. Okay, here's another picture. This is a computer representation. Okay, so what is prominent there are the helices, no? And it's color coded. It's probably a multi-subunit protein. Okay, so one subunit is here color coded yellow, the other subunit is color coded blue, and then it's also superimposed to the wire stick model. No? So instead of showing the spherical atoms, they're just zigzag lines. Again, why is that the case? Depends on what they want to emphasize. So let's look at the prevalence of alpha helix in proteins. The alpha helices can actually vary from very few percent to almost 100%. It will depend on the protein. Could there be factors that disrupt the alpha helix? We just came from describing the ideal structure of the alpha helix. Of course, in reality, the presence of the side chains will also matter. One very prominent amino acid is proline. Recall that it has a cyclic structure, okay, therefore has hindered rotation. In addition, when the nitrogen already participates in the amide formation with the next amino acid residue, then there's no longer any hydrogen available to participate in hydrogen bonding. So once you see proline as part of a chain in your primary structure of proteins, you can safely guess that most likely that section of the protein does not have an alpha helix secondary structure. What would it be? Well, we cannot really predict, but you can say that most likely it is not an alpha helix. Others could be electrostatic repulsion. Okay? Recall that when we described the alpha helix, we said that in a turn, the nearest neighbor will be about 4 residues away. So check whether every 4 residues you have similarly charged side chains, meaning they're both positive or they're both negative. So the electrostatic repulsion would probably repel and destabilize your alpha helix at that section if it occurs. Okay, so you can safely guess if you were to predict and say that is this section most likely an alpha helix? This is your check. Every 4 residues they have the same charge, so most likely it will not be an alpha helix. Another one would be steric repulsion. So, okay, so recall that again, the beta carbon, well, again, a reminder, what is the beta carbon? If this is your alpha carbon, this is the beta carbon, no? And then not shown in the structure of proline is the carboxylic acid. Okay. Okay. So steric repulsion will occur for valine, isoleucine, threonine, or just the, anyway, the large amino acids. So again, if they are in close proximity, then most likely it will destabilize your alpha helix. Okay, so let's move to the description of the beta sheets. It's an extended polypeptide backbone, as you can see on the reduced view on the upper left-hand corner of your screen. No? So I'll show a close-up of that in just a short while. But again, in contrast to the alpha helix, the beta sheet is a more extended polypeptide backbone. Also, notice the hydrogen bond that's formed, okay, the pattern. As shown in the picture, the polypeptide chains run from in the horizontal direction, and then the hydrogen bonds are in the vertical direction, more or less. So it is described that these hydrogen bonds are perpendicular, quote unquote, to the polypeptide chain directions. Also, in contrast to the alpha helix, wherein all the hydrogen bonds are intra-chain, the hydrogen bonding pattern in beta sheets could be intra-chain or inter-chain. Okay? So if it's intra-chain, the polypeptide chain folds back to itself. Okay? So intra-chain, meaning, okay, if this is the polypeptide chain, this probably just like turns around and then continues in this direction. Okay? If it's interchain, then if this is the direction of your polypeptide chain, then it probably like moves around, okay, and then continues on, and then you form that hydrogen bonding. So that's interchain. And then there are two types. The two polypeptide chains that form the hydrogen bonds can either be parallel or anti-parallel. So let's look at a close-up view of that figure. Okay, to emphasize. Okay, so this is a bigger picture. As we can see, let's look at the polypeptide chain. Again, this is the nitrogen, the alpha carbon, the carbonyl carbon, nitrogen, alpha carbon, carbonyl carbon, and so on. So if we look at this chain, we can say that this is the N-terminal position, and this is the C-terminal position. Now, if we look at the other polypeptide chain to which it is hydrogen bonded to, let's look at again the nitrogen. This is the nitrogen, alpha carbon, carbonyl carbon, nitrogen, alpha carbon, carbonyl carbon, and so on. So if we look at these two chains that form the hydrogen bonds with each other, they're both running in the same direction. So we can say that these two polypeptide chains are parallel in direction. The bottom one, okay, if you look at this nitrogen, this is the alpha carbon, this is the carbonyl carbon, nitrogen, alpha carbon, carbonyl carbon. So the top chain runs from N-terminal on the left to the C-terminal on the right. For the bottom, okay, you have the nitrogen, alpha carbon, carbonyl carbon, nitrogen, alpha carbon, carbonyl carbon. So the bottom chain runs from left to right, N-terminal to the C-terminal. So these two chains run in opposite directions. The term that is being used is anti-parallel. Okay, so notice that in the anti-parallel scheme, the hydrogen bonds are perpendicular to each other. In the parallel, they're to a certain degree, but if we take the average of these two hydrogen bonds, then they will be perpendicular to the direction of the beta sheets. So let's ask, why is it called a beta pleated sheet? Okay, so imagine now the peptide planes. So recall the peptide planes, and if they do that, okay, they can be, you can sort of like place the peptide planes in this corrugated or pleated sheet. So it's in the imagination of the people who were trying to describe the structure of the beta sheets. Okay, so they look like pleats. So hence it's called the beta pleated sheet. And then why alpha and why beta? Well, the alpha was the first one that was discovered, and then beta was the second one that was discovered. So if probably there's a third regular pattern in the backbone chain of the polypeptide, then it will probably be something called be gamma. Okay, but so far there's only two that have been regularly found. Okay. Now, once again, okay, we've described ideal situations, but just like anything, if there's something that's regular or ideal, okay, nothing is perfect. There will be some irregularities. So what are they? Okay, so other structures are the 310 helix. Sorry, I don't have a picture of that one. It has like 3 residues per turn and 10 atoms in a ring forming the hydrogen bond. There's the existence of the 2,7 and the 4.410 helices. There's also a beta bulge, which allows a change in direction of the beta sheets. So here they are. So uh, these are pictures of beta sheets also. Okay, so if you look at this classic bulge, what happens? Okay, so there's almost a one-to-one correspondence between the amino acids on one chain and the other forming a hydrogen bond, and then it skipped one in this illustration. So what happens is that there's now a change in direction of the beta sheets. There are others like a G1 bulge and a wide bulge. We don't really need to examine their structure fully unless you probably go into master's degree and you're focusing on biochemistry. Okay, so for us, it's sufficient to know that again there can be irregularities in the regular structures. One of the effects of these irregularities is a change in direction of the beta sheets. Okay. Another thing that often happens is the reverse turns. Okay, so if you imagine once again the polypeptide chain, it does not really occur in one straight line. It has to change direction some way. We've already seen how a change in direction could be achieved by the bulges in the beta sheets. But what if it's not beta sheets? Okay, well, there's also the reverse turns. Okay, so the polypeptide chain needs to change direction. Glycine and proline are most often found in these reverse turns. Okay, glycine because it has the flexibility. The side chain is also a hydrogen, so it's small. There's no hindrance. There's no steric hindrance to its rotation. Proline, well, proline has a built-in turn, so it's often found in reverse turns. Okay, so here's a picture. There's two types of reverse turns. Okay, type 1, okay, the side chain is outside the loop. Okay, so let's number the amino acids involved in the turn. Okay, so this is the rest of the chain, the rest of the chain. Let's just count this. This is residue number 1 in the turn, residue number 2 in the turn, residue number 3 in the turn, residue number 4 in the turn. Okay, so in type 1, the side chains are outside the loop. Okay, well, all of them have to be outside the loop because they take up space. But specifically, type 1, the third residue, we're focusing on the third residue. Okay, so in this case, because all the side chains are outside the loop, any amino acid can be in the third position. There are no restrictions. For type 2, okay, the side chain is inside the loop, and only glycine is small enough to occupy this space inside. No? So, or okay, it can also be assisted by the built-in turn by proline, but still the third amino acid has to be glycine. Okay, why is that? Okay, so let's look at the side chain of glycine. So compare the orientation of this alpha carbon for glycine to the alpha carbon in a type 1 turn. Do you see the difference? Here, the orange ball and the white sphere, which represents hydrogen, face outside the direction of the turn. Here, if this is the direction of the turn, notice that again, the hydrogen for the amino acid and the hydrogen specifying that it is glycine is pointed towards us, and this is the direction of the turn. Okay, so this would have been the side chains. If this were a regular amino acid or non-glycine amino acid, there would be too much steric hindrance for it to form the turn. Okay, recall that again, this is the ball and stick model. If this were a space filling model, there's really not much space inside over here. No, there's too much. So in other words, it's only glycine that can fit into this space. Okay. Before we proceed, okay, may I ask you to touch your skin? Okay, so manipulate it. Is it elastic? Is it tough? Massage your muscles. Okay, do it. And also be aware of your connective tissues. I also want you to imagine your bones too. Okay, done? So let's proceed. Okay, the reason I asked you to do that exercise is because we're now going to discuss the collagen triple helix. Collagen is a component of bone and connective tissue, and it's the most abundant protein in vertebrates. They form water-insoluble fibers. Okay, so again, your skin doesn't dissolve. Part of that reason is it's composed of fibers. And it also has great strength. No? So when I asked you to pinch your skin, it did not flake off or fall off. Okay, you'll be in serious trouble if that happened. Okay, so here's now the structure of the collagen polypeptide. Okay, the one on the left. This is just one chain. It's probably reminiscent of the alpha helix, except that again, this is not the alpha helix. The general structure is a repeating sequence of proline and glycine, or X-hydroxyproline glycine. Okay, so I think recall that again when we were introducing the common amino acids, we said there were uncommon amino acids, and one of the uncommon amino acids cited was hydroxyproline. So proline and hydroxyproline together make up 30% of the residues. You can also infer that about 30% of the residues would be glycine. Another common amino acid, well, another commonly found uncommon amino acid in collagen is hydroxylysine. Okay, so what makes this triple helix? So if you now have these three chains winding around each other, you're going to get the collagen triple helix, also known as tropocollagen. Each of the helix twists around each other, and every third residue would be oriented towards the center of the helix. Ooh, once again, I need you to use your imagination and look and imagine the space filling model. Okay, so if there were three chains winding around each other and every third residue is oriented inwards, what's the amino acid that can fit in? That's right. It's only glycine that can fit in. So you can probably now understand why every third residue is glycine. And the proline, the presence of the proline, uh, uh, introduces a built-in turn, okay, within the chain. No? So this proline, there's already a built-in turn present. This intertwining of the polypeptides makes it strong. Okay, so I want you to imagine, for example, why rope is twisted. Okay, why can't it just be a simple string? No, well, the intertwining of those individual strands makes your rope strong, and in this case, it makes the collagen strong. Okay, and then the strands are held together by hydrogen bonding, okay, involving hydroxyproline or hydroxylysine. So here now are the three strands winding around each other. Okay, they're just color coded. So the yellow strand, the blue strand, and the purple strand. Each of these represents this chain. No? So, and this is now also in the space filling model. And then the tropocollagen forms links with other tropocollagen, and they're cross-linked with other tropocollagen using lysine and histidine residues. The amount of this cross-linking increases with increasing age. That's why you may have noticed that, for example, if you're eating meat, and then you may have gotten the comment that, matanda na yung baka. No? That means we usually want to eat like the softer muscle, the softer meat comes from the younger animals that have been slaughtered. Okay. And okay, so here let's just complete the picture showing the interlinking of the tropocollagen with other tropocollagen units. No? So this cross-linking is the one that increases with age. So the younger you are, okay, the less cross-linking that will be observed, and then the older you are, the more cross-linking. Now, an interesting aspect is the role of vitamin C with collagen. Okay, it's needed by the enzyme that hydroxylates proline. Hydroxyproline, as mentioned earlier, stabilizes collagen structure by hydrogen bonding. Deficiency results in a disease called scurvy. Okay, so here's a picture of scurvy, results in like bleeding gums. And while I was searching for bleeding gums, actually, or scurvy, this picture also came up. No? So notice the hair on the skin is not what we expect normal hair to look like. So again, that's the role of vitamin C. Okay. Before we go to the tertiary structure, okay, well, I won't be asking you any exercises, but we'll just look at super secondary structures. Okay. With here, we mean common combinations of alpha helices and beta sheets. Examples, the beta-alpha-beta unit. So this is very common in proteins, that a beta sheet section is connected to an alpha helix, which is connected to a beta sheet. So it's a beta-alpha-beta unit. Common also will be an alpha-alpha unit, meaning a section is alpha helix, and then the next few amino acid residues is not really an alpha helix, and then after that will be another alpha helix. There can also be a beta meander. Okay, with a beta meander, it just means that again you change the direction of your polypeptide chain with different beta sheets. A Greek key, okay, maybe better with an illustration. Okay, so here they are. Okay, so on the upper left is beta, a linker peptide chain, the alpha helix, and the beta. Also, the alpha unit. Okay, so notice that this is not an alpha helix over here. And then a beta meander. What do you do when you're meandering? Okay, you're just wandering aimlessly. There's no specific direction. Okay, so this beta meander, a beta sheet that goes up, beta sheet that goes down, beta sheet that goes up, beta sheet that goes down, beta sheet that goes up. Okay, reminds you of a Sex Bomb song, right? Okay, I know you're singing it. Now, another one is a Greek key. Okay, so if you're not familiar with Greek motifs or architecture, okay, or the key, there's something like this is a common architectural motif. Okay, so if you look at the buildings and some rooms, they may have decorations that look like this. No? So so they describe this as being a Greek key. Okay, so this flow of beta sheets looks similar to this one, so it's a Greek key. There could be others, of course. These are just examples, and these are commonly repeated in different proteins. Okay, so you have the different motifs. Okay, so these are also have the repetitive supersecondary structures. Okay, so here's the others now. So notice that this is repeated in the same polypeptide chain. Uh, I think this is also called like a beta barrel, okay, because again, in your imagination, it looks like a barrel. So there's a barrel in this section of the polypeptide chain, another barrel in this section of the polypeptide chain, another barrel in this section of the polypeptide chain, and so on. So there are many ways by which the polypeptide chains like have conformations in a protein. Okay, and then remember that this is only a section of a protein. Now, this is not the whole protein itself, but only a section of the protein. Okay, so before we go to the tertiary structure, let's just state that there are two types of protein conformations. They can either be fibrous, okay, so almost self-explanatory. The polypeptide chain are made up of long fibers, and they will be water insoluble. They're usually strong, okay. Their examples are collagen, okay. We've already described the structure of collagen and the occurrence of collagen. Later, we will look at fibrin and keratin, okay. So fibrin, component of the web and the silk, and keratin, okay, touch your fingernails and your hair. The other type of protein conformation is globular. Okay, these proteins are spherical in shape, globular, spherical, synonymous to each other, and they are water soluble. Just going about fibrous proteins, their functions are mostly structural in nature. Okay. Okay, so just a picture of examples now. So a filament, this one is slightly different. It's a 4 right-handed twisted protofilaments. Earlier we talked about collagen, just made up of 3. Myoglobin, we will look at myoglobin in just a short while.